Today is our speaker joined us. Ladies and gentlemen, please be attentive. Rohit sir, are you there?
Yes, sir. Yes, sir. Yes, sir.
सर को पोस्ट बनाइए. Very good evening, sir. Dr. Amit sir, are you able to hear me? Sorry. रोहित सर को पोस्ट बनाइए प्लीज.
डन सर, बना दिया सर.
बना दिए.
यस सर.
सर प्लीज अनम्यूट योरसेल्फ.
Yeah, thank you.
वेरी
Hello.
गुड इवनिंग.
Good evening.
वेलकम behalf of Dr. Prakriti ma'am and the Agriculture Valley Research Foundation team. Very welcome, sir, on our platform.
Am I audible?
Yes, sir, you are audible, sir.
Okay.
सर विद इन 2 to 3 minutes स्टार्ट करते हैं सर.
Okay, please let me know.
ओके सर. रोहित सर प्लीज सर का हेल्प कीजिए पीपीटी शेयर करने.
सर तब तक पीपीटी एक बार शेयर कर लेते हैं ना, जब तक सेशन शुरू होगा. अमित सर. हेलो अमित सर. अच्छा सर, मेरी आवाज आ रही है आपको?
जी मैं बात कर रहा हूँ. हेलो डॉक्टर अमित सर, आर यू देयर?
Yes, I am there. I'm here. I'm here. Tell me.
सर तब स्क्रीन शेयर कर लेते हैं सर, तब तक जब
हाँ,
तक
मैं कर
सेशन
रहा
शुरू
हूँ.
होगा.
मैं कर रहा हूँ.
ओके ओके.
हेलो.
सर इसको पीपीटी आपका स्क्रीन दिख रहा है सर, एक बार इसको
हाँ.
पीपीटी मोड में कर दीजिए सर.
पीपीटी को क्या करना है?
पीपीटी मोड में करना सर इसको. हाँ सर, यहाँ पे. यस सर. सर एक दो स्लाइड्स आप प्लीज मूव करेंगे, लाइक फॉर कंफर्मेशन की स्लाइड्स. हाँ मूव हो रही हैं सर. फाइन फाइन सर, ग्रेट.
Is that okay?
यस सर, फाइन. थैंक यू सर.
सईदा मैम, आर यू देयर?
Yes, I'm here.
प्लीज स्टार्ट.
Okay. Am I audible?
यस मैम, यू आर ऑडिबल.
Good evening, everyone. A very warm welcome to all of you joining us today for this insightful online session organized by the Agriculture Valley Research Foundation, New Delhi. We are truly delighted to have an esteemed gathering of researchers, academicians, and students with us today, united by a shared vision of advancing sustainable agriculture, innovation, and scientific research. I am Saida Nahal Zehra, Technical Secretary to the founder of AWRF, serving as the program coordinator for today's session. Today's session focuses on the theme of agriculture for sustainable development and a step towards innovation in crop science. In an era where climate change, soil degradation, sustainable food production are major global challenges, such discussions become extremely important for shaping the future of agriculture. It is now my great honor and privilege to introduce today's distinguished speaker, Dr. Amit Chandra Kharkwal, Director and Professor at the Amity Institute of Microbial Technology, Amity University, UP. Dr. Karakwal is a highly respected scientist with over 22 years of experience in plant microbe interactions, fungal biotechnology, and sustainable agriculture. His research expertise includes root endophytes, biofertilizers, biocontrol agents, and climate resilient agricultural technologies, all of which are crucial for the future of sustainable farming systems. He has an outstanding academic record with over 120 scientific publications, 29 patents filed, including 8 granted patents, and even a US patent to his credit. His impactful work has not remained confined to just laboratories. He has successfully transferred 3 microbial technologies to 8 companies, leading to real world applications that have benefited over 1 million farmers. Dr. Karakwal has received research support from prestigious organizations, including the DBT, GST, ICAR, DRDO, as well as from the Ministry of Environment and Forestry. His innovations have earned recognition at both national and international levels, including awards from the FICCI and the Institute of Food Technologists and Grow Further. With this remarkable background of contributions to science, innovation, and farmer welfare, it is truly an honor to welcome him. So, without any further delay, I now request Dr. Amit Karakwal to kindly take us through his valuable insights and address this session. Sir, if you would, the virtual platform is all yours now. Thank you.
So thank you, Miss Saida Nahal Jara, and thank you, Dr. Pragati, for inviting me for this important session. I hope I am audible and visible to you, all of you.
Yes,
So
sir. Perfectly.
I am going to talk about my work, which is mostly translational work centered around novel plant growth promoting root endophyte with agricultural applications for climate resilient sustainable agriculture, which is, quite in demand nowadays, more so because of the turbulent global situation. Even our Honorable Prime Minister has requested in his address that to use more of the natural fertilizers and reduce the dependency on, chemical fertilizers because of the obvious increase in the fuel cost and a lot of other matters. So this has become all the more important. So what we start from our hallmark work, Memoirs of Rutonic. So this book is downloadable. If you just search it, Rutonic PDF, you will get it from Google. And this is discovery that is made by Professor Dr. Ajit Verma, the founder director of Amity Institute of Microbial Technology, from where I from where I'm speaking right now. And he is no more with us. Last year he left his for his heavenly abode, but he inspired us to do this work further. So Rutonic, a magic fungus, and what we say in Hindi, biokhad, jadon ki takat. So it has tremendous role in agriculture, horticulture, and forest productivity. So if you look at the origin and discovery of Plutonic, you see this desert, Thar desert of Western Rajasthan, where there is hardly any water to grow. And from the roots of these plants, this fungus was isolated. And here you see this fungus is isolated from the roots of these plants, and it is forming the rhythmic ring in a solid medium and the ball-like structures in the liquid medium, right? And if you see, this fungus has tremendous role in enhancing growth promotion in various crops, including, for example, turmeric, right? Sugarcane, here you can see, right? And here you see the ratoon crop of the sugarcane, how it is being treated. And here you see just a minute, I think there is some issue in my PPT. Let me check it again, because I don't I think I'll just shut it down. Just a minute, please bear with me because
Sir,
yeah, I have not. Yeah,
it's there, sir. Yes.
it's there.
Yes, sir.
Okay. It's okay. I don't know, but something has happened, and yeah, okay. Right. Okay. So I'll just start it again. Uh, okay. So this I was talking about the rutonic, the magic fungus. Okay. So just wanted to give a brief background of the rutonic first. So rutonic is basically an example of a mycorrhizal symbiosis which was not earlier observed. So I hope you know what is a mycorrhiza. Mycorrhiza are the fungi which are intimately associated with the root system, and that is why they are named mycorrhiza. Myco means the fungus and the rhiza means root. But these are obligate symbionts. They do not live outside the root of the plant. They are unable to survive. And after some time, if they are not getting their host, they are, they are unable to survive in the soil system. And particularly in the conventional agricultural scenario where we are using chemical fertilizers and toxic pesticides, insecticides, etc., so the mycorrhizal survival in the soil becomes all the more challenging. So this mycorrhiza comes in various shapes and size. If you see here, mycorrhizal spores, and the mycorrhiza, because they are unable to survive, they do not multiply and die, despite they were discovered somewhere around in 1886 by German scientist Frank. No one has made pure culture of this mycorrhizal fungi, and there we come with the Piriformospora indica, or now more commonly known as Serendipita indica. We have branded it as Rutonic for our internal usage, and International Symbiosis Society called it Champion of Symbiosis and Discovery of Millennium during the Boston Conference. So this is from where I was coming. The discovery took place in the Thar Desert of Western Rajasthan. This is how it looks, it grows. And yeah, so this is the model organism, the electron micrograph of this fungus. And because is the cursor, are you able to see the cursor? Hello?
Yes, sir. Yes, sir.
Okay, very good. So if you see, this is a pear-shaped spore, right? So that is why it was earlier named Piriformospora indica. And this discovery prompted the global scientific community to develop an entirely new order in the class Basidiomycota, Sebacinales. And this was then further renamed as Serendipita indica, and the spores are autofluorescing under UV light, and they are pear shaped. So Piriformospora, pear shaped forming spore, indica discovered from India. So that is the whole background. The genome of this fungus has been partially sequenced by Alga Jugaro group in 2011, and the current name is Serendipita indica also. So this is how it looks like. So we have used this fungus and promoted in various states of India, including Punjab, Himachal Pradesh, Haryana, Gujarat, Karnataka, Delhi NCR, and Rajasthan, etc. And what we have observed over a period of time, having multiple properties, plant promoter, biofertilizer, bioregulator, right? So all these properties of this fungus are published, right? So it belongs to the class Basidiomycota, right? And now what we are going to talk about its application. So when I am going to show you the plant promotion activity, so there is only one thing common: control versus treated. All other conditions are same. So treated means the plant treated with the spores of this fungus, right? Control means not treated with the spores of the fungus. So control versus P. indica, as you see in the GMAs, right? So we have tested over 148 plants in our greenhouse and field, and the interaction was found positive in terms of growth. So looking closer, if you see in this confocal microscopy picture, these green colored P. indica spores, right? Here they are colonizing the cortical region of the, this plant section, the stele, outside the xylem and phloem. So here they colonize, and you see here they are sending the fine hyphae outwards, okay, which helps in its various plant growth promoting activity. And the fungus remains alive only in the new roots of the plant, right? It's not entering into the older root system. So this is how we multiply in a fermenter, and then, we formulate this. And how do we apply this? This is a talc-based formulation, for example. This is one of the example, but you can make multiple formulation, granular formulation, liquid formulation. There are multiple options, and the best treatment is seed treatment, but you can also do seedling treatment. You can also do soil application. You can also do the application with the irrigated water, the fertigation. So the purpose is to mix it with the seed or the seedling, or apply to the soil and allow it to early colonization after the plant germinates, the seed germinates, right? So the early colonization of the fungus by the root does the magic. What is the magic? I am going to show you, right? So now, apart from promoting plant growth, which I have shown you in the example of 148 plants, what it does, it promotes early flowering. So this is the model plant tobacco, where you can see the early flowering as a response to the treatment, right? At the same time, okay. This is another plant, the Brassica napus, where you see the fruit setting is complete in the treated plant, whereas there is flowering just initiated and some fruiting in the control plant. So this is how, it stimulates early flowering in the plant, right? So what is the advantage of this? I'll come to it later. And here you see the medicinal plant Coleus vauxhallii. Now, yeah, not only early flowering, but it not only promotes growth promotion of medicinal plants, but also enhances their active ingredient. So this is the Bacopa plant growing in the minimal medium. So in the plant inoculated at the same time, right? The one inoculated with P. indica, they grew faster. Not only they grew faster as compared to control, but there was 20-fold enhancement in antioxidant production, 7.5-fold enhancement in antioxidant activity, and 3-fold enhancement in the active ingredient by bacosides. So this is what the value addition it does to the medicinal plant. So medicinal plant, there is a lot of demand for natural herbs. So there it helps in increasing their value, productivity and value. This another plant, the anthole and fenchone in essential oil production in Funiculum vulgare, what you call SOF in Hindi, right? So they were announced. This work was shown by our Iranian collaborators. And Stevia rebaudiana. So everybody is aware about Stevia rebaudiana, the source of natural sweetener. So, you see the growth of the control versus treated, but not only growth, the stevioside and rebaudioside content was also enhanced in these plants and published. Another plant, Artemisia annua, source of anti-malarial drug, artemisinin. So here you see the difference between the control and treated. But not only control and treated, other plant parameters were enhanced, right? And most importantly, if you see the last column, artemisinin, right, content is increasing nearly twofold here if you see. So the value addition of the medicinal plant, as I said, this is field trial of Spilanthes calva in central India, source of medicinal compound spilanthol, again enhanced growth as compared to the control. This is Vidania somnifera, again enhanced growth and more alkaloid production in Vidania somnifera. This is Chlorophytum borevillianum, Safed Musli, very medicinal plant, very high in demand. So this plant was cultivated through tissue culture, plant tissue culture technique, but there was problem in the establishment. With the treatment of the fungus, it took, over 90% plants survive in the field, tissue culture raised plant survive in the field, and this survival was coined, this type of transplantation was coined by Professor Verma at that time as biological hardening. In tissue culture, we do the hardening, but this type of adaptability was coined as biological hardening with the help of beneficial microbes. Now, this is field trial of turmeric. Here you see the turmeric plants, not only there was growth enhancement, but also enhancement in volatile oil and curcumin content of the plant. Aloe vera, here you see micropropagated aloe vera plants, not only bigger in size, but here you see the gel content, right? 16.36% enhancement in gel and aloin content 52%, the secondary metabolite. Here you can see the even the after the hardened plants, you can see the difference in the growth. Similarly, this Ajadirachta indica AFRI, here you see growth promotion in neem as compared to control in Plantago ovata, isabgol also. Here you can see, and up closer you can see the maturation, early flowering and maturation. So it is completing the life cycle early, enhancing the product value, right, and helping the farmer to bring the produce early to the market. So from medicinal plants, we come to the agro-horticulture, and we have standardized with our talc-based formulation having viable spore count of 5 into 10 to power 5 to 7 spores or CFU, colony forming unit per gram. So across multiple varieties of crops, okay, we have standardized how much seed treatment has to be done among different crops, and maximum seed treatment that is there is in potato, wheat and sugarcane, that is 1 kilogram per acre. So this basically helps in this way in enhancing the productivity of the crop and giving multiple other attributes we will see here one by one. So if you see this field trial of sugarcane that was done in collaboration with PAU Ludhiana and the current VC, Dr. Sohal, he performed this experiment, right? And if you see in the ratoon crop, not only the agronomic parameters were enhanced, but the sugar content also enhanced as compared to control, right? So this is where it is important that per plant biomass in ratoon crop, the sugar content enhanced. And if here you see the control versus treated plant, okay. So what was found that, the absorption of micronutrient like iron and copper enhanced significantly as compared to control. So it helps in the biological solubilization of nutrients, leading to enhanced isolation of the nutrients which are otherwise locked in the soil. It's not that soil is deficient in the nutrient, but these nutrients are not solubilized for and bioavailable for the plants to absorb. So that is one another aspect where this fungus plays an important role. Now, this is a field trial which was done in cotton in Punjab, and where we observed over 20% increase in yield and reduction in pesticide usage by over 50% as compared to control, right? And another field trial which we did in Punjab. All these trials which I am talking about, they were done in collaboration with the Agriculture Department of Punjab government. So all these are authenticated, verified trials. 22% increase in yield in potato. In brinjal, if you can see right from the beginning, right between treated and control, and treated plants were also resistant to sucking pest and whitefly. That was a feedback given to us by the farmer, right? And early flowering, early fruiting as a consequence of that, and 20% yield enhancement was observed. Similarly, this work we did in collaboration with PAU again, right? And here what we did that, there was no decrease in wheat production at 75% chemical fertilizer input. So with reducing the fertilizer input by 25%, there was no compromise in the yield. And at 100%, yield was increased to 11%. So another trial in the farmers field in Mulampur district in Ludhiana, Punjab, here you see early maturation in the treated plants, and various growth parameters were enhanced, but most importantly, the yield was enhanced by 45% in the farmers field. This trial we did in Delhi. You can see the crop growth between the control and the treated, right? Higher tillering in the treated, and but we could not get the yield estimate from this Delhi trial, right? But another similar trial from the district gene, there the farmer reported a 20% yield enhancement here, and you can see very well what is the reason for that. Pronounced biomass, right? Root and shoot architecture between control and treated, you can see. And this is not one-off thing. I will show you more such cases. Now, this field trial was done in Punjab, right, in the district Mohali across 5 locations by the ADO, right, and they have shared with us the report and what they reported to us, right. The 14.64% enhancement in maize production was recorded across 5 trials. And similarly, we also did the same trial under DST seed project under rain-fed condition in the village Noorpur Bedi in district Ropar, Punjab. So this was a rain-fed condition. That means no irrigation available, right? So here you can see the difference between the control and the treatment in the crop size. And again, roughly 15% enhancement in the yield. Not only yield, but treatment showed early flowering and increased pest tolerance, that is resistant to black spot disease and pest flies as the feedback given to us by the farmer, right? Similarly, in P, 8.5% enhancement in yield was recorded. In rapeseed mustard, in the outskirts of Delhi, 12.5% yield was enhancement in yield was recorded. And similarly, this again in the rain-fed area, this trial was done, same area. Now here we were surprised to see 49.17% enhancement in yield, and alternaria black spot infection was found less in treatment as compared to control, right? And seeds obtained from treatment were found to be good in texture and color. How good, you will see yourself, right? First, you see the eye-level photograph of control and treatment, right? And you can see. But most importantly, you see while the flowering is still on in the control, the treated seed is full of pods. Not only this, the stand, you look at the stand of the person standing of the same height, And here, if you see the 5 plant biomass, right, randomly selected, right, here you see the biomass of the 5 crops, right. Not only this, here you see the roots of the control and treated and the seeds, right. So the difference are apparent, and please note this is done without irrigation, without even a single cycle of irrigation in the rain-fed area. Similarly, in that same area, another crop, linum, ulci, that is linseed, 21.9% yield enhancement was observed. And then we come to another important crop, that is guar, right? Field trial was done and 16% increase in guar seed production. So as a consequence of this result, right, I was invited by an industry, Sunita Hydrocolite Private Limited, as you can see, and here you see I am addressing over 2,000 farmers, and this company had a buyback arrangement with the farmers, and to all the farmers they provided 4 kg of seeds plus 100 kg of our root tonic, right? As you can see here in this box, we have powder, lot of powder, formulated powder, and we are applying to the guar seeds and which were sown. So it reached the masses in that way. Now from the yield enhancement, not only does yield enhancement, but I have mentioned it also helps in the disease resistance. So biological control of disease. This one example in the Balachor district of Punjab, here you see, these are two contrasting 4-acre field of bottle gourd standing next to each other. And here you can see the yellow versus green. Only difference is our root tonic intervention, just one single application and 33.3% increase in yield and reduction in pesticide usage. So the commercial pesticide at that time, Coragen, which was applied once in a week in the treated field, here in the control, it was applied every 3rd day to control the disease. So you can see still, right, the disease control, right? So it enhances the resistance of the plant against the pathogens. Similarly, here you see on quinoa in the normal plant, early flowering and fruiting, so more yield for the farmers, right? But these are some trees which were infected with the virus, and the farmer was about to, take it out, and we, requested the farmer to leave just 5 trees. So the 4 trees we applied the fertilizer, and 1 tree was left without the fertilizer, and you can see here the picture. So we could even recover the virus infected plant, and it started flowering and started fruiting. So that is, it prevents the infection of virus also. So up close you see between minus and plus P indica that suppression of disease is taking place, right? And there are multiple reasons, and for that a lot of publications have been made. If you have any questions, I will be happy to answer you later. Now, most importantly, this fungus, which I told you, is discovered from the cold desert of Rajasthan. And to our surprise, it worked equally well in the cold desert of Leh and Ladakh. So this is a poly-inch greenhouse, right, developed by the DRDO scientist. And in the month of November itself, the seeds are sown, right? And as the season get warmer, the seeds germinate, and then the plants are transplanted outside. As a consequence of our application, the seeds germinated early, the plants grew better, developed better, and here you can see between the control and treated, even the head is not formed, here the head is matured, right? Similarly in beetroot and cauliflower again. So this, what advantage does this give? This expands the window of cultivation in regions like Leh Ladakh, where there is limited time for cultivation. So instead of one crop, the farmer can start thinking about taking two crops also. So that is tolerance to cold stress. So from now this discovery, we come to the another discovery, that is another fungus, that is HNB9, a novel biological agent for agriculture. But before I start with HNB9, may I ask if any of you have any questions, right? So we can take some few questions here regarding Rutonic. Hello, Mr.
Hello.
Yeah.
Yes, sir.
If you
Questions
have,
will be asked at the end, sir. Sir.
okay, okay.
Initial,
So
yes,
yeah, because I was speaking in flow, right? So I
yes.
thought that if you have any questions, we can ask. Okay, fine. So let me come to the our next innovation, right? So this is HNB9, a novel bioagent for agriculture. So Rutonic, discovered by Professor Dr. Ajit Verma and developed at MIT. Then Teloromyces albobiverticillus, formerly it was designated as Purpurogenous species also for some time. So this is HNB9 is a strain of Teloromyces albobiverticillus fungus. That is a patented azenically cultivated novel root colonizing fungal strain. Now we are first to demonstrate that species of Teloromyces can be plant growth promoting and root colonizing. So colonization of plant root by fungus results in pronounced growth enhancement and resistance against various stress that I will show you. And fungus has exhibited various plant growth promoting PGP activities like phosphate, zinc and silica solubilization, ammonia and auxin production. In addition, fungus also secretes a red color compound. Okay, whether it is a compound or compounds, just right now a method of investigation, matter of investigation. So that is secreted extracellularly in the medium, right? It can be used for various industrial uses, including food, pharma, and textile industry. So we are validating that. This is a publication of this fungus published in Nature Scientific Report. Now, this is how it looks like. So here, this is a fungus, benign, growing on various fungal culture media, right? And up close here, you see zinc solubilization, phosphate solubilization, silica solubilization, and IAA production. When I say solubilization, these are insoluble compounds which are being solubilized with the help of this fungus. Not only this, we have confirmed its non-mycotoxin producing nature by testing it on the standard creatine sucrose agar medium. So all my friends who are working on fungi must check their species if they have discovered first on this medium to ascertain the production of mycotoxin. So if you are producing mycotoxin, you will get this response. That means you stop the work here. Do not take this fungus, further because it is harmful for human and animal health. That is very important test for doing basic test for selecting non-mycotoxin producing fungi. So this is a non-mycotoxin producing fungi. Right? This is how it looks like. Right? So conidiophores are biverticillate. So that is why elbow biverticillus and globose to ellipsoidal in nature spores. And this is the confocal image of conidia, stipes, conidiophores, and ascomata producing ascospores. And this is the confocal image, okay, of the spores and the mycelia that is emerging from the spore, right? So these mycelia, they basically penetrate the roots and then colonize the roots. This is the scanning electron microscopic figures, right? So fungus is well characterized and its phylogeny was done, right? Molecular characterization, why it is HNB9 spore. So this is also important, my friends, who are working in the identification of new species, that you need to do multi-gene phylogeny. So we use the known molecular markers to have a widespread genomic coverage. So ITS, BEN-A, SCAM, and RPB2 genes, right? These are conserved genes whose markers are well known. So this is how we did the bioinformatic analysis, and we did the pairwise identity of these 4 genes, right? So based on this analysis, we were able to see the similarity of the HNB9 ITS contig, BEN-A contig contiguous sequence, right? And RPB2 and CAM, right? 4 genes, and we were able to establish their relationship with the closely related species. And if you see T. rubrifaciens and T. albobiverticillus, they are shown the more closely related species. And a later paper also proved that rubrifaciens and albobiverticillus are one and the same species. So that is why we named it albobiverticillus. So with the consensus, right, we basically named the species. Now I come to the important plant part, that is its ability to enhance plant growth. So first of all, again, my friends, look clearly that you have to do the seedling toxicity test. The fungus should not be toxic to the seedling, right, before you take it further. So it was not toxic to the seedling, and then we went to the greenhouse. For example, maize and okra, here you see. So enhanced shoot biomass and root biomass, both were observed in greenhouse condition. And similar is the story to the Rutonic, you can see, right? And this is up close how the formulated powder is made. So this is what I was telling you about the red color production, right? Extracellular pigments. These pigments are playing very important role that I will show you later, right? So this is our first trial which we performed in Yamuna Khadar region in Delhi NCR, that is opposite Mayur Vihar on P, right? So what we observed, early flowering and early fruiting and pronounced, yield enhancement by the farmers in the farmers field. And this is, the spores of the fungus, you can see colonizing the root of the plant, right? So microscopy shows us this is how the spores live inside the roots of the plant. Okay. Now, this is a trial which was again done in collaboration with the scientists from PAU. And here what you can see, right, the not only the enhancement in agronomic parameters, but across 3 cultivars of wheat, right, as compared to control, there was 13% enhancement in yield, right? Significant enhancement in yield, right? And similarly, here you see in the rice, again in the farmer's field, right? 33% yield enhancement just by application of this talc formulation once. All other conditions are similar. Now, in this case, we are not just limiting to the one fungus. We have developed its various consortium with other plant growth promoting microorganisms. So this is the first consortium we have developed with the microbe that is known as Bacillus subtilis, right? Bacillus subtilis has become very famous these days because that is the state microbe of Kerala. So before it became the state microbe of Kerala, we got this microbe from NAIMCC, right, MOU, and we interacted with this fungus. Here you see, right, and here you can see both are living with each other. So both are compatible with each other. So my friends, I will just take 1 minute more explaining that when the fungus is interacting positively with bacteria, right? Beneficial fungus interacting beneficially with bacteria, the combination becomes more beneficial because a bacteria gets a matrix support, right? So, as we say, highways are the path to prosperity. So, does the fungal hyphae are the path to prosperity? Because in the highway, the vehicle moves, and in the fungal hyphae, the bacterium moves. So, I have just, used this corollary. Hyphae and highway, both are important. So here you see in greenhouse, we did some trial, right? And root length, fresh weight, dry weight, and total chlorophyll content increased. Here you can see in the picture, right? In the consortium and the microbes alone, and root colonization also increased significantly, right? By the fungus, right? So here you can see the root colonizing, right? And more pronounced colonization of root took place, right? And we developed this consortia, right? In the same medium, where the fungus and bacteria in the broth, they were co-cultivated, right? And what we observed that the hyphal swelling took place in the fungal hyphae, and as a result of that, there was more pronounced release of spores, right? So what we want in a biofertilizer, large number of spores. So that is what this consortium is giving us, right? And not only the spore number, but the spore size also increased significantly, spore count also increased significantly, right? And this talc-based formulation was prepared, right, and applied in the farmer's field. So here, what we did, the rice nursery was ready. So we made a slurry of the formulation and we used the seedling dip. So in the slurry, we will dip the roots for 20 minutes, right, and then transplant it. Okay, so this is the only treatment what we gave, right? So here my student working at that time. So yield enhancement controlled at 18.34 quintal per acre, HNV9 26.26, and with Bacillus subtilis 29.64. So you can see, right, the consortia enhances the maximum production. So now, not only we just did the growth promotion, but we also looked for its ability to, look at the drought stress, right? 100% irrigation with then control with treatment and T3, where you say you see the growth parameters are increasing in different irrigation regime, 100%, 50% you see, right? Irrigation again, and restricted irrigation, that means no irrigation. So there was significant enhancement in the growth in restricted irrigation and 50% irrigation. And the same was, tested in field, right, on wheat crop and at PAU, right? Here you can see what is most important here, my friend. If you see the grain yield in the full irrigation uninoculated is 48%. In restricted irrigation, it is 53%. Now, what is restricted irrigation in the field, right? Restricted irrigation is that in Punjab, there are 6 cycles of irrigation, and here we have skipped 3 cycles. So only 3 cycles of irrigation we have taken, right? And the yield is more than uninoculated of full irrigation. Of course, in rain-fed, yield is significantly low, but if you see effective tillers is showing the same trend, but if you see in the rain-fed, the 1000 grain weight, right, it is not getting compromised. It is similar, right, to uninoculated control, right? So that means the grain weight is not compromised even in rain-fed conditions. So what we look for, we look for the consortia, PGP activity, and what we found that the silicon solubilization index enhanced significantly in consortium as compared to, right, without. The Bacillus subtilis is not solubilizing silica at all. HNB9 is solubilizing silica, but together they are solubilizing silica more, right? So consortium has a synergistic activity. Now, another consortium which we made was with Enterobacter cloacae, right? Enterobacter cloacae SK4 strain we isolated from the coal mines of Sikkim, right? And this tulanomyces is a very strong phosphate solubilizer. How strong it is, I will show you in the coming slide. And this bacteria Enterobacter cloacae demonstrate for not only phosphate solubilization, but ammonia production and potassium solubilization also. So potassium is not at all solubilized by the HNB9 strain, but solubilized by Enterobacter cloacae. So here you see again the positive interaction was first established in a dual co-culture plate assay, and this is how Enterobacter cloacae, ammonia and production was quantified. And here you see fungus is not solubilizing the potassium in Alexandrov media. Bacteria is solubilizing potassium, but look here, fungus plus bacteria. The fungal hyphae, the bacteria is positively interacting, now solubilizing more ammonia, more potassium as compared to bacteria alone. So that is the beauty of consortium. So, how this system is working, just to quickly see. Soil nutrients, they are locked in mineral forms. Microbial consortium release organic acids and metabolites. Solubilization of phosphate, potassium along with zinc and silica. Ammonium oxide production enhances root development and nutrient uptake. Now, if you see what is the economic advantage with this reduced chemical fertilizer when we do. So typical chemical fertilizer anywhere in the Indian setting will cost somewhere around 2790 per acre. That is average cost we have done. If you do 50% chemical fertilizer plus bio NPK, your costing will come somewhere around 1696 per acre, and estimated fertilizer saving will be 1100 rupees. Bio NPK dose, if you assume at 1 kilogram per acre, right? Now, if you look at return on investment, observed yield enhancement can be anywhere between 15 to 40%, right? So if you take baseline crop revenue of 40,000 per acre, then additional revenue of 6,000 to 16,000. So the total farmer benefit will be there between 7,000 to 17,000. This in addition of enhancing their soil fertility and enhancing the quality of the crop, the quality of the produce, right? Also. So we provide the market opportunity to the global biofertilizer market that is valued in billions and growing rapidly. Okay. So as I told you in the beginning, there is pressure to reduce chemical fertilizer usage, strong demand for biological and sustainable agriculture input. So that is where our bio NPK platform that is aligning with the next generation biological agriculture. And that is why we are sitting here and discussing. So we can, integrate with the industry. Okay, we can complement conventional fertilizers such as urea, DAP, and MOP. This fertilizer you can, coat with urea and apply to the field. Okay, we can have integration with existing fertilizer distribution networks. Okay, there is opportunity for formulation development in field validation, and one can achieve a scalable microbial fermentation process, right? Now, if you compare the bio NPK with chemical NPK, right? So the nutrient delivery in chemical is direct chemical salts, where here we are doing biological mobilization of soil nutrients. Nutrient efficiency is moderate. It is improved here. Soil health, it can degrade soil microbiome. It can enhance microbial activity. Environmental impact, leaching and runoff risk in water bodies. Lower environmental footprint, application frequency, multiple applications. Here, lower dose with biological persistence in the soil. Value proposition, it is a commodity fertilizer, it is a high value biological input, right? So, key idea here is chemical NPK supplies nutrient, bio NPK plus activates nutrients already present in soil while improving plant nutrient uptake. So, I told you separately how strong phosphate it is, right? When I say it's a strong phosphate solubilizer, how strong, right? It is very strong phosphate solubilizer, right? If you compare, we have achieved a TCP bioacetate fermentation process. TCP is tricalcium phosphate and it can solubilize 40 gram per liter in a fermenter, right? With controlled fermentation. Now, if you compare it with the rock phosphate soil application or rock phosphate fermentation, right? The rock phosphate application is slow and variable phosphate release in the soil. Rock phosphate fermentation is having a limited industrial scalability, but we have the scalability in our hand and we can provide in a single microbial product soluble phosphate plus microbial inoculant, right? Okay, so our TCP fermentation provides engineered biological phosphate. So that is TCP bioassisted for fermentation platform where we have used tricalcium phosphate as a fermentation substrate by resolution using our fungus by having a defined concentration window for efficient mineral processing and simultaneous generation of soluble phosphorus and fungal biomass. This is how it looks like. At 4 gram in 100 ml, there is a clear suspension that is made and the calcium phosphate mineral particles, they form a dispersed system stabilization as observed by zeta potential analyzer, right? So if you again compare the costing here, right? Rock phosphate, TCP and triple soluble phosphate chemical fertilizer, right? Look at their cost, okay? P2O5 content comparable with the chemical. Biological solubilization is 60 to 100% it achieves. It is already soluble. Effective cost per kg available phosphorus will be 130 to 60, slightly higher than that. But process suitability is a fermentation process. It's a chemical process altogether different, but much better than rock phosphate. So rock phosphate will appear cheap per ton, but TCP fermentation delivers cheaper usable phosphorus due to higher biological conversion efficiency. So the rock phosphate cost will be less, but efficiency if TCP fermentation is very higher. Similarly, we have made a nano zinc bioformulation where we use the two microbes together, Teloromyces and Enterobacter, and co-cultivate it with the zinc oxide nanoparticles. What we have observed, right, using the scanning electron microscopy and zeta potential analyzer, right, the zinc oxide nanoparticles, they attach to the fungal bacterial consortium and remain suspended in the broth also. So we can use both the pellet fraction for plant growth promotion and supernatant fraction as a nano zinc oxide suspension, right? So it enhances soil stability because this consortium is neutralizing the charge of this fungus bacterium to near neutral, right? And also making available zinc oxide nanoparticles of the size 99 nanometer. So zinc is already approved for the zinc oxide nanoparticle are already approved for the agricultural application. And here we are talking about the biologically stabilized zinc oxide nanoparticles. So we have done the plant growth efficacy of the consortium in the wheat, right? You can see it is promoting the plant growth as compared to alone and control, right? And in fenugreek also we have done, right? Here you can see. And this bacteria that is isolated from the coal mine of Sikkim. So we have done the arsenic tolerance test again. And this bacteria tolerates up to 10,000 ppm of arsenic. And the fungus is also tolerating over 300 ppm. So we are looking into exploring the aspect of bioremediation of arsenic. Arsenic is a major problem in the eastern part of our country, and we are very sure, right? We are performing some certain studies that this consortium will be very helpful in the remediation of arsenic from the agricultural field, right? Now, another consortium which we have made, this is the first time we have made in the world this type of consortium where we have 3 protozoa and 3 microbes, 1 fungus and 2 bacterium together, right? And what they do, they feed on the fungus and bacteria, right? And as they feed, they induce selection pressure. So this is a carrot and stick policy in a simple term, right? They induce the stress on the microbes, the protozoa induce stress, and microbes multiply more, and the protozoa which eats the microbe that releases the lot of nutrients in the soil, right? So if here you see protozoa with HNP9, right? Another microbe, right? Here you can see more phosphate solubilization as fungus alone, right? Zinc solubilization, higher auxin production, higher IA production, higher biofilm production, right? So this protozoa was, this consortium was applied in wheat, right? In fenugreek, here you can see how it is promoting the growth, right? So this again is a work in progress, the protozoa, the formulation. And now we come to the another property of this fungus, if I have the time, biocontrol activity. So this is biocontrol of Fusarium oxysporum, as you can see, right, in this picture, the left and right. The fungus culture filtrate is having the biocontrol property, right? And within 16 days, you can see it is completely breaking down the fungal mycelia. And on the right side, if you see this pane, this is the inhibition of Fusarium race 1 by the fungus. This is a very important, pathogen of banana. Then you see in fenugreek, biocontrol of Fusarium, right? So we have a DBT project running, right, where we have shown, right, using the consortium of Fusarium with the, sorry, HNV9 and Enterobacter, there is pronounced, suppression of Fusarium oxysporum across the 3 former species, Lycopersicki, Shalt, and Vasinfectum. Okay. So, dual culture led to 38 to 40% inhibition as compared to the microbes alone. Not only this, we confirmed this by Evans blue staining the dead microbes, as you can see, right? And I'm not going to detail. And then we performed the plant microbe interaction. Here you see this is infected with Fusarium, all 3, right? And Fusarium infection is very severe. The plants are stunted as compared to control. But where you have microbe, okay, whether SK4, HNB9, or combination of both, right, they are able to overcome the infection of Fusarium oxysporum, right? So here you see percentage root colonization by Fusarium in the treatment. T2 you see Fusarium 75, 90, 94, right? And as the consortium is applied, it is between 10 to 15%. So that is what we know by biological control of Fusarium oxysporum, right? So the percent disease index is going down significantly as compared to the control, right? So we are able to, make this consortium as a biofertilizer, biopesticide, okay, upscale production in 800 ml flask, you know, 1200 ml flask, right? And from here, we have optimized the consortium, okay, with the inoculum volume, what is the inoculum density, which we are going to keep for the production of both the viable spore count of this fungus plus bacteria. And we are able to take this production using picovascular medium in a 3 and 7 liter fermenter also. This is 3-liter fermenter, right? So we observed the various growth and biochemical response with increase in number of leaves, roots, and shoot, as you have seen, higher chlorophyll content, reduced SOT, CAT, catalase that is, and ascorbate peroxidase enzymes and DPPH activities indicating stress mitigation, and enhanced NPK and micronutrient content in root, shoots, and rhizosphere soil. So here you can see growth parameters enhanced as compared to fusarium and the consortium, right? In fenugreek, okra, tomato, all, right? Chlorophyll content, you see significant increase in chlorophyll content, right? Antioxidant enzyme activity, you can see the fusarium enhances the antioxidant. The consortium is bringing down the antioxidant enzyme activity, right? DPPH activity, right? In various crops, and micronutrient content, nitrogen in fenugreek, okra, tomato, phosphorus, right? In all three, and potassium, so significantly increasing, right? Zinc and iron content is also getting increased, right? Soil potassium, soil nitrogen, phosphorus. When we talk about soil, that is rhizosphere soil zinc and iron content, okay, is increasing. So consortium treatment improved the soil enzymes activity and soil respiration and increased alkaline phosphatase enzyme. And the fungus produces the hydrolytic enzymes, okay. Fusarium spores in the soil reduced, okay. And developed consortium was re-isolated from soil after treatment. So we confirmed the presence of the consortium in the soil even after the treatment. Okay, so these are the soil enzymes. Okay, the micro enhancement in the soil enzymes means increase in the soil microbial activity, right? That is an indicator of soil microbial activity, which is going down in the fusarium infected plant in the soil as compared to control. And with the introduction of this microbial consortium, the soil enzyme activity is further enhancing. So these are the hydrolytic enzymes which are produced by the fungus alone, right? And the plate count inhibition of fusarium was done. So fusarium was reduced significantly in the soil after the treatment. Okay, and this is very important because it is very, very difficult to treat the fusarium infected soils, right? We did the field trials and culture filtrate of this fungus intervention also. Now, this is a unique field trial where the field A is control, non-infected field. Field B is fusarium infected field where there is no treatment. And C is fusarium infected field with treatment. So here it is very important for you to see that is what happens in the fusarium infected field, right? It is very difficult to control the infection in the fusarium infected field. So the plants simply did not survive and they were required to be taken out, right? As compared to fusarium infected field with treatment, our microbial treatment, the plants were able to establish, right? This is again this ongoing work with the culture filtrate, right? Again, the culture filtrate of the fungus is also inhibiting the fusarium oxysporum, right? And this is the work is ongoing, and we have isolated certain metabolites and their major classes. Right now, we have classified their coumarins, flavonoids, anthracenones, and terpenoids, right? So this work is going on. And another work which we have done, interaction with entomopathogenic fungi, Metarhizium and Bavaria. Now, both Metarhizium and Bavaria bassiana, they are well-known fungi and entomopathogenic fungi for the biocontrol of insect pests. So together here is a list of insects which Metarhizium and Bavaria are, inhibitors of, right? So what we thought of, as a master student working with me, that makes some consortium of these fungi. So what we observed, compatible interaction between Talaromyces and Metarhizium and Bavaria bassiana, right? But what happens when Metarhizium interacts with the Bavaria bassiana, right? Here you see Bavaria bassiana is mycoparasitic on Metarhizium, right? So that is the thing. What we observed that lysis of Metarhizium hyphae by the Bavaria bassiana. To our surprise, when we co-inoculated HNB9, right, this mycoparasitism was inhibited. And not only this was inhibited, even these three fungi together, you can see in the confocal microscopy, they are happily, living with each other. And when they are applied to the sugar cane, right, the 15% yield enhancement, and farmer reported us biocontrol of white grubs and nematodes, right? But when we use the culture filtrate of this fungus, HNB9, this also made neutral, interaction between Metarhizium and Bavaria, and there was no lysis of the hyphae of Metarhizium, right? Even the culture filtrate extract, okay, the pure compound which we are talking about, right, initiated the positive interaction. Now we are looking at which pure compound among the culture filtrate is responsible for this, right? That is work in progress. So what we can make, right? We can make a soil-based biopesticide and a biopesticide combining Metarhizium and Bavaria bassiana for aerial spray. So we can have both the options of open, right? So this I have explained. As for the root tonic, again, we have standardized for the various crops, okay, with the seed for the seed treatment, talc formulation, for seedling treatment, and for the soil application also, right? Seedling 500 gram per acre, soil application 1 kilogram per acre. Now, as a spin-off of this work, right, we made an antimicrobial fabric, right? This is a normal fabric, and this is the fabric that is colored by this the filtrate of this fungus, right? And this is having very strong activity against the E. coli, pathogenic E. coli and Staphylococcus aureus strain as per Japanese standards. Now, this work has got different recognition. The Bavaria bassiana Metarhizium consortium, which I showed you, right? This is transferred to the to the industry also in Noida. HNB9 is also transferred to the industry alone, right? This work featured one of my students pitched this idea in the Falling Wall Labs India certificate of participation in 2021 we got, right? This technology was 15 brilliant young minds from India, right? 15 top technologies from India, they were selected, right? This is my interview that is there in the Grow Further. We were listed as one of the, globally 6 centers of regenerative agriculture for our work in Rotonic and HNB9, right? So this is how it looks like. Okay, this is what the clipping said in the website. Okay, this is most recent, the FICCI Bharat R&D Summit among some 60 technologies from various fields, from AI, from engineering, from science, chemical engineering, other sciences. Okay, this technology was given the first award, right, as the technology in that summit. Okay, this is the certificate of recognition. So we provide various licensing and collaboration opportunity for our multiple technologies, including Rotonic and HNB9. The technologies which I have shown you are protected through patent filings. We have opportunities for pilot trials and industrial scale-up through mutual agreements. Possible models can be technology licensing, joint development, or strategic partnership. Goal is commercialization of next generation biological nutrient platform. So I acknowledge DST, DBT, ICL Israel, our founder, honorable founder president, our chancellor and vice chancellor, ma'am, okay, for allowing me to do this work. And these are my team members. Of course, Professor Verma is no more with us, but he will always remain my mentor. Then there are various faculty members who are working with me, various brilliant students who worked with me or working with me, and they shaped this journey very well. So thank you very much for your patience listening. Thank you once again.
Thank you so much, sir, for such an interesting session. Now, if anyone has any questions or doubts, they are welcome to unmute their mic to ask the question directly, or they can drop the same in the chat box. In the meantime, sir, I would request you to wait 2 minutes as majority prefers to actually write in the chat box.
Okay, so please read if I am unable to read from the chat box and read the
Of course,
questions.
sir,
So
no
I will
issue.
be answering it.
Participants, please either unmute the mics for your questions or please write it on the chat box. Sir, we have from Mr. Ajay Sharma. He is thanking you for such an informative presentation. He's asking, I would like to ask the given increasing frequency of the climate-induced stresses, how do root endophytes perform under drought, salinity, and heat stress conditions compared to conventional PGPR, plant growth promoting rhizobacteria?
Okay, so a very good question. And as I have shown you some work on the drought or limited water conditions, and basically the endophytes, they are basically colonizing the root. So they are inside the root. So that itself is a protected environment for them, right? And they send their fine hyphae outwards, okay? And as I've shown you, they release the organic acids and enzymes which help in the stress tolerance, as I've shown you in the drought, better water absorbing activity. And similarly, P. indica, for example, has shown, enhanced salinity tolerance at a high salt stress like 50, 100, even 150 ppm of the salt stress by upregulation of the plant, tolerant genes also. So, and if I say how it compares, it's not a question of comparison. It is a question of having multiple attributes in a single organism. That is what we are talking about. So, there is nothing conventional. This is also a plant growth promoting microorganism like HNB9 or Rutonic, and other microbes like Bacillus or Bacillus subtilis or Pseudomonas fluorescens. So they are, but only difference is different classes. So the fungal bacterial consortium, we are using the PGPR, right? Rhizobacteria in the consortium with the fungus, right? So that enhances the uh, ability of bacteria more because the bacteria get a solid matrix support, right? So consortium alone, consortium works better than alone, right? Either it's bacterium or the fungus. So that is not the question that how it compares with the conventional. Yeah, but it comes with certain added advantage, right? So whether it's drought, salinity, and heat stress. Heat stress, what we have got feedback from the farmers field, right? Uh, in 2022 and 23, I think 2 years in consequence, there was heavy heat stress in the month of May when the grain setting takes place in wheat. The farmers reported to us that our treatment, resulted in better grain setting as compared to control. So that's a matter of more investigation, right? So that we'll be doing, and we are happy to collaborate also. So I hope I answered your question, right? So
Thank you so much, sir.
we're not looking for comparison, but we are looking for collaboration, making consortium. Thank you.
Thank you so much, sir. We have another question from Abby Sam. How
Yeah.
can we treat the fusarium infected
Now,
fields?
fusarium infected field, you can treat the roots of the plant at a time of sowing, right? That is one way. And another way is soil application, right? Okay. So you can have different types of soil application like flood irrigation or other conventional treatment. And fusarium infected field, I have myself observed it gives much better result. Okay, if applied at the right time, and one can complement with other strategies also. But yes, it gives very good result. Okay, so soil application or seedling dip treatment or seed treatment, okay, or fertigation. So in multiple ways, you can look for the seed application, right? This fusarium biocontrol. Okay, yeah. So next question.
Thank you so much, sir. Many are actually praising for your presentation. So thank you so much again for that interesting presentation. We have a question from Miss Ruchika. How can we improve the damping of infected fields, sir?
Okay, damping of disease. As of now, I have not worked on, but I think some of my student, they have reported to me the biocontrol of Sclerotina rosei. So I hope that it will work in the damping of condition, right? Basically, you see what is happening, that biocontrol is happening by two ways. By one is the fungus is releasing the enzyme, but the other is most important element that is part of our earth crust is silica. 23% silica is present in the earth crust, right? And it is so unfortunate today that what we see the advertisement of silica fertilizer, root soil application and foliar spray of silica. Now that is what is present most in the soil is now being applied from the outside. No, that is most unfortunate. But the silica absorption helps in lot of protection, multiple disease protection just by application of silica, right?
Yes,
Okay.
sir.
So how can we treated seeds without using chemical? You bring the chemical coated seed. No, no, not an issue. Okay. We will just coat the seed with our fertilizer. What we need, our fertilizer is having spore count of 5 into 10 to the power 5 to 7 CFU per gram. What we need, even a fraction of that, just colonizing the roots, that is enough for us. Okay. Yeah. Then you have what regulations governs, of course, the FCO and the Biostimulants Act. Okay. Fertilizer Control Order. So we pitch it as a phosphate solubilizing fungal biofertilizer, HNB9. But the regulations sometimes, unfortunately or fortunately, you may say, no, they are not keeping up pace with the science. What is the fun of publishing so many papers, publication, when they are not seeing the light of the day in terms of their actual field applications? No?
Thank you so much, sir. We also have another question from Subha. Very useful and informative presentation, sir. Can you suggest any remedy for false smut in paddy and YMV in pulses?
Okay, so I have not worked on the false smut. Maybe you can share some more information on the false smut, because I am not a pathologist, right? Yeah, but it will definitely have more, better result than than without any other treatment, or with in combination with other treatment also, right? And what is YMV, yellow mosaic virus? Definitely, with our experience in Rotonic, it will control the YMV impulses.
Thank you so much, sir. I believe that's the last of our questions. So if there aren't any more questions, I think we will conclude our session. So, as we come to the end of this highly enriching and insightful session, it is my privilege to propose the vote of thanks on behalf of everyone present here. First and foremost, I extend our deepest gratitude to our esteemed speaker, Dr. Amit Chandra Kharkwal, for sharing his invaluable knowledge, experience, and visionary insights. Your remarkable work and practical contributions towards transforming agriculture and benefiting millions of farmers are truly inspiring for all of us, sir. Thank you. We are sincerely thankful to the Agriculture Valley Research Foundation and all the collaborating institutions for organizing this impactful session and providing us with such a valuable platform for learning and interaction. I would also like to thank the technical and organizing team for ensuring the smooth conduct of this webinar. And also, thank you to all the participants for your active participation throughout the session. With that, we conclude today's program, hoping to continue such insightful interactions in the future. Thank you, everyone. Thank you once again, sir, and have a great evening ahead. We conclude today's session. You are all free to leave the meeting now. Thank you.
Thank you very much. So may I
Thank you so
leave
much,
now?
sir. Yes, sir.
Thank you. Thank you so much.
Once
Thank
again,
you.
sir.